Baldwin-Brown, and I'm going to tell you about some genomics work that I've been doing in the clam shrimp Ulanania texana. So here's a clam shrimp. It looks like that. That's a male, and that's a hermaphrodite. You can tell because the male has these little claspers that are hard to see there. And then the females, well, hermaphrodites have these little egg pouches on their back. So what got me interested in clam shrimp was that there are a lot of problems that come with working with certain model organisms for genetics, which these guys tend to solve. They have a lot of the same advantages of things like flies, where they have a smallish genome, shortish life cycle, but they have a great advantage that being vernal pool shrimp, they lay eggs that go into diapause for indefinite periods of time. So they live in these pools that have water only part of the year, and when the water runs out, they dry up, all the adults die, but the eggs live on and wait until water returns, and then grow into new clam shrimp. So I'm hoping that this in the future can be a model system that we can use for genetics that will allow us to archive old populations rather than having the problem of populations having to be maintained forever or die out. So I got excited about these guys, and then realized no one's ever done genomics on these in any form. And so that was going to be a challenge. So I realized, is this my turn? Oh, there we go. If I was going to work with these guys, I would need to make them into something resembling a model organism. So there are a few things you kind of have to have in order to do good genomics work, right? You need a good assembly. You need it annotated in some form so you can tell what genes you're looking at. And then you'd like to know a little something about the populations. So I took it upon myself to generate a decent de novo genome assembly, and then annotate it myself, and then at the end of this talk, this is still ongoing, but I'll talk a little bit about the work in identifying differentiation of wild populations that we've looked at. So the first thing that we realized was that we needed the genome assembly. In the past, doing high-quality genome assemblies was really expensive and difficult, and doing low-quality ones, they tended to be very low-quality but pretty cheap. But in the last few years, PacBio sequencing has become pretty common and pretty inexpensive. So we went about developing a new PacBio genome assembly pipeline that would be a little bit more cost-effective than what's been done in the past. It turns out people have been using exclusively PacBio data to generate really high-quality assemblies that often get entire chromosomes as a single contig. But it's pretty expensive. It takes like more than 100x of PacBio data a lot of the time. So we realized that there are hybrid assemblers that exist that use Illumina and PacBio data, and there are these PacBio-only pipelines that exist that use only PacBio data. And both of them give all right assemblies under certain conditions, but we realized that there's a funny overlap that I'll show you in a second between them that makes it clear that if you could merge a hybrid assembly and a PacBio-only assembly, you could produce an even higher quality assembly. So that's what we did. We made our own merging program, and we merged a hybrid and PacBio-only assembly together and then polished it up to make a very high quality assembly. We actually first tested this using public Drosophila data so that we could test and make sure our method was working and not producing terrible misassembled assemblies. So we're pretty confident it works well. Here's our test data set, right? These are hybrid assemblies with different levels of PacBio coverage. And then this is a measure of the contiguity of the assembly, NG50. You can see that it gets pretty good and then kind of tops off. And then this blue line is the PacBio-only assembly. It starts off pretty terrible, but then gets quite great. But when you merge the two assemblies, this orange line, you can do much better with much lower levels of sequencing coverage. So we took that in mind, got some PacBio and Illumina sequencing coverage in the clam shrimp, and assembled the clam shrimp genome, and we got an NG50 we're pretty happy with. It's even above the line that we predicted. So I think it's a pretty good assembly, and I think we have good reasons to believe that it's a correct assembly. You can see here's this is a cumulative coverage plot of the genome. Each of these points represents a contig. So this is a 40 megabase contig, and then the next biggest contig is about 20 megabases, and if you add them all up, you get the genome size approximately, which is about 125 or so megabases. So we're pretty happy with the assembly. Then we needed to go ahead and annotate it in order to actually use it for anything, right? So we did some RNA-seq. I'm not going to talk a lot about the RNA-seq because the exciting parts of it I think are still ongoing, but to keep it short, we did RNA-seq on males and hermaphrodites, and we did an annotation with Trinity and Augustus, which is a gene predictor program. And we're pretty happy, I think, with the genes that we found. And then we're planning, although we haven't completed this analysis yet, to compare the male and hermaphrodite, because the clam shrimps have an interesting property that they have males and hermaphrodites, but no true females, the way that a lot of, or at least some plants do. So we're hoping to identify regions of the genome that are overexpressed or underexpressed in males or hermaphrodites. But that's still ongoing. So we have a pretty good genome. We have a bunch of annotations. These are all our Trinity transcripts, and then those actually come down to, I think, 18,000 genes or something when you run it through Augustus. So we're pretty happy with that. Now, we get to the exciting part, which is all the wild populations that we sequenced. So now we have this assembly, and we wanted to do something with it. One, to show that our assembly was working, and two, because we wanted to learn something about these cool species. So we got our collaborator to go out in the desert in New Mexico and Arizona and collect samples from a bunch of populations. What you do is you just dig up some dirt, take it back to the lab, and then add water to it, grow up the shrimp, and then grind them up and sequence them. And the way we did it was we sequenced 100 individuals from each of the populations by just grinding them all up together, doing pool-seq. So we pool-seqed a bunch of these populations, and then we did some basic population genetics calculations on them. First, we made a quick, this is just like an H-plus tree made in R. We did a quick tree to show how the populations is the populations that are close to each other, like these guys, which are all very close to each other, are all related to each other. That actually goes against a very common belief in the vernal pool community, which is that these vernal pools are pretty isolated, right? There aren't a lot of ways for a shrimp to travel from one pool to another. But one way or another, they are, in fact, getting around. Because if you look at this pair of populations, WAL and EE, EE is directly descended from the WAL population. That's a lab population that we sequenced alongside these guys as a control. And it's only 6 generations diverged from WAL. So you can see they have a degree of differentiation that matches some of these WAL populations that happen to be right near each other. Here's JD1 and JT4. You can see they're practically the same population. So in a way, it was a shame to find that out when we did the sequencing, but it also revealed a truth about vernal pools that I think people hadn't been aware of before. So we took these guys and we wanted to calculate some other basic population genetics facts, right? So one thing we did was there are a bunch of programs now, one in particular is LDX, that's what we used, that can take Illumina data and calculate LD over short distances for you. The way it does it is it looks for regions in the genome, or it looks at individual reads, paired reads, that either share 2 alleles or have 2 of the opposite allele. Any given pair of reads must come from a single individual. So you can say with some certainty that, how did I do that? Oh, there we go, we're back. So you can say with some certainty that that those 2 alleles are associated on a chromosome. So you can calculate LD over short distances, and we did, and we used that to extrapolate out to longer distances. Whether or not you believe that is up to you, but we did it. We also calculated the autocorrelation of standard deviations of allele frequencies in the neighborhood around SNPs. So you look at how autocorrelated the standard deviations are from one SNP to its nearest neighbor, next nearest neighbor, and so on. And what you can see is it looks like there's not a ton of LD beyond the first few SNPs. So it kind of indicates that if you're going to look for a single selection, you probably want to look for small numbers of SNPs in small windows, or maybe just a single SNP. So we did that, and then we decided to, there's some of the population statistics. So we did that, and then we wanted to actually start looking at differentiation of the populations. This part is still ongoing. I still have things running on our cluster back in Irvine, so I can't tell you too much about this, but I'll leave you with some of our first analyses. So the first thing that we did was compare the WAL population, which I already told you about, to EE, which is its descendant in the lab. in order to show that those two populations were not really differentiated from each other. And what we did was we did a chi-square test comparing YL to EE, and then we QQ plotted it against the p-values that you expect from the chi-square test. And indeed, you find that there's it's pretty linear, right? There's not a lot of signal of differentiation of the populations. There's a bit, right? That it doesn't stay 100% on line. But compare that to two of the wild populations plotted against each other. This is LTER versus Tango 11, which were our two highest coverage populations. And you can see there's a huge signal of differentiation between them, which is probably due to differences in ancestry. So we ran those QQ plots. We were pretty happy with the fact that these two populations seemed like they were pretty much related. So we went ahead and pooled them together for the remainder of the population differentiation statistics. So we pooled them together, and then this is what's still going on now. We're running a few different statistics trying to compare the populations in order to identify signals of differentiation between populations that are localized to small regions of the genome, which you might think are signals of selection. We don't know for sure that anything is right now, but I'll show you. I think we have a few results that look fairly convincing. So this is maybe the most powerful statistical test for this kind of thing out there right now. The Bayenv, Bayenv is a program written by the Coop Lab, which can generate several different statistics. It's usually made to work with Bayes factors, but here I'm showing the XTX population differentiation statistic. We're also running Bayes factor analyses against environmental variables that we know, but that's still ongoing. So what I saw here when I saw this was it looks like it's all kind of random, right? There are some areas that kind of look like peaks, like that one, but it doesn't look that great. But this is just looking at individual SNPs. Our coverage is not that great. It's about 50x per population. So we went ahead and ran a sliding window, a 5-SNP sliding window, and it kind of changed the whole thing, right? Immediately you can see there are certain peaks that actually look pretty nice. Oop, I'm losing the laser pointer. There we go. So peaks like that look pretty good, peaks like that look pretty good. I went ahead and highlighted a few. I don't think you should take away from this that I necessarily believe these yet, but I think this is an exciting thing that's happening, and I wanted to share it with you guys. I'm hoping that we'll be able to validate some of these SNPs and show that they really do look like they have some kind of selective influence, but that's an ongoing process. So for conclusions, if you have your own pet organism that you want to work on, and it's not a model organism, you can still do high-quality genomics on it with not that much money. And